Hello friends, welcome again, and in this session we are going to discuss about the recombinant DNA technology and its applications. How DNA is engineered in vitro, how DNA is engineered in from your DNA of interest. ठीक है? One DNA is of your DNA of interest. So why there is a need of doing recombination of DNA. So for example, if you want to synthesize, if you want specific protein, for instance, you can take example of your pharmaceutical product. So if you want any protein, then it naturally it is produced, but it is produced in very less quantity, and you want to increase the production, and you want to increase the output, then you have to make recombinant DNA, so that gene responsible for production of that particular protein is isolated, that is inserted into the vector, and the vector is finally transformed into the uh, cells, for instance bacterial cells, and then you can multiply the bacteria, grow, ferment that bacterial suspension. You can convert that bacterial suspension into final product, and there is isolation and downstream processing for the final collection of that product, desired product. Okay, so recombinant DNA technology, we will see few recombinant DNA technology and its few applications. So here you can see in the first image, that is biotechnology recombinant DNA technology application. First one is a dolly and its surrogate mom. Okay, so dolly and its surrogate mom. Then second, in the second image you can see genetically modified rice. Okay, so this was the first this dolly was the first transgenic animal. Okay, you, you have to remember, this dolly was the first transgenic animal. Actually this was the question asked in Niper. So there are two favorite questions from Niper, that is the first one is dolly and second one is this golden rice. Okay, so genetically modified rice, which is also known as this rice is also known as a golden rice. So it is a golden rice, it is enriched with vitamin A. Normally there is no vitamin A, or if there is it is, if it is present in very less amount, so in white rice. Okay, so scientists have identified gene responsible for production of that vitamin A. The isolated gene, then modified, genetically modified the gene, the inserted gene into vector, and then vector infected the rice, and you will get a with golden rice with high amount of vitamin A. Okay, so these are the dolly, that is first transgenic animal. This golden rice with uh, vitamin A content, and this is a embryonic stem cells. Embryonic stem cells, they are used in gene therapy. So these are nothing but the three applications of recombinant DNA technology or genetic engineering. Okay, so biotechnology defined as broadly, it is the engineering of organism useful for our purpose. Okay, so we humans are very selfish. We do each and everything for our sake of good. So we are very selfish. We do genetic engineering, we do fermentation, we do your farming for only for our purposes. Okay, so biotechnology involves the creation of hybrid genes and their introduction into organism in which some or all genes is not normally present. Okay, so if any organism is lacking the gene, you have to correct that gene using outside gene. For that you have to prepare hybrid gene. So we will say how we can prepare hybrid gene, then what is the steps, etc., etc., in the next slide. So I just want to say, show you that this is a genetically engineered or recombinant DNA technology product, that is known as salmon fish. Okay, you can see the big fish, that is salmon fish, and the you can see in the left side, that is salmon fish. You can see in the right side, that is original fish. So salmon or genetically engineered salmon fish, you are able to see in the left side. So that is a big, and this fish, salmon fish, is why it is engineered to get the fish. Why we eat fish? So you will have more meat content in the fish, that genetically engineered fish. Okay, so this was the few applications. So what is biotechnology? The use of living cells to make products such as pharmaceuticals, then foods and beverages. Okay, then use of microorganisms such as bacteria to protect the environment. For example, genetically engineered bacteria. Such bacteria will digest the your digest your compost, your wastes. They can digest your waste. So presently a lot of waste uh, are there in the seaside, etc, etc. So to digest that waste, you can use genetically engineered bacteria. Then the use of DNA science for the production of product, diagnostic and research. So it is also one of the applications of recombinant DNA technology. So what are the some applications of recombinant DNA technology? So use of bacteria, yeast and plants can be modified. Bacteria, yeast, plants can be modified to produce important pharmaceuticals, enriched foods, vitamin enriched foods, then industrial products. So it is nothing but the use of it is possible by use of recombinant DNA technology. For example, this monoclonal antibody is produced using recombinant DNA technology. The first you can see in the first, that Synges. This is a monoclonal antibody produced by recombinant DNA technology. Okay. Then second, here you can see Novalin. It is a insulin produced using recombinant DNA technology. Okay. This is nothing but the genetic library. Okay. So you can see this corn is a genetically modified corn. So these are the some few applications. We will see what are the more applications. For example, recombinant DNA technology are used for animal cloning. You can see in the first image that horse is exactly cloned for the daughter. Means the daughter and horse is looking exactly same. It is nothing but the clone of each other. Okay. So and then your stem cell therapy for various genetically mediated diseases. For example, genetic diseases are there, hereditary diseases are there. So you can use these stem cells using recombinant technology. These stem cells are used to treat their genetic disorders or diseases. Then DNA fingerprinting is there. So for that is mostly used in the uh, forensic science. DNA fingerprinting is mostly used in forensic science to identify the accused. Okay. Then we also have genetically modified crops and foods. Then gene cloning for pharmaceutical production, that is one of the most important application of biotechnology and recombinant DNA technology. Then gene therapy, that is also one of the important application of biotechnology and your recombinant DNA technology. Okay, so animal cloning. So, it was the first animal cloned, that is known as Dolly. Okay, Dolly and its surrogate mother. So genetically identified pigs have been cloned. So why there is need of cloning, doing these all things? So we can answer the basic biology, genetics and DNA fingerprinting, etc., etc. So to answer the basic questions of biology and for herd improvement means you can improve your herd, your cultivation of animals. Okay, herd means kalap. Marathi, in the Marathi it is known as kalap, to increase the herd genetic purity, etc. For pharmaceutical production, then to satisfy our desire. For example, pet cloning. If you have done if you have isolated DNA of pet, you can regenerate. If the pet is died, then you can regenerate that exactly the same looking pet. It is for the, so how Dolly is made. Okay, so First of all, dolly is a combination of two ships. First one is a white ship and second one is a black pastry ship. So if you have to, they have isolated Udder cells. Udder cells means the cells of mammary gland. Okay, so Udder cells you can see from white ship. Udder cells are isolated. Then from that cell DNA is isolated. That DNA is injected into the egg cells, isolated from black head ships. Okay, so black pastry ship and white pastry ship. Combination of these two DNA will result in the dolly. That is known as dolly. Okay, it was the first transgenic animal, dolly. It was the first transgenic animal, and it is important for Niper because they have asked 2-3 times question on this transgenic animal, first transgenic animal. So dolly is the first. So some of people believes that they are firm believers of DNA technology, while other people consider it as a stunt. But it is not like that. So attempt also have been made to clone the humans, but it is highly unlikely. It is not. It is unfavorable in our profession and scientific community. So gene cloning for pharmaceutical production, for example, insulin. Most of the people nowadays are suffering from type 2 diabetes, and for their treatment, insulin is required, and it is obtained most of the time. It is obtained from recombinant DNA technology. Okay, so what is exactly recombinant DNA? So consider you have one DNA or gene. So you have only single DNA. The manipulation and combination of DNA from two sources. For example, your DNA and other person DNA. If it is combined, that will result in recombinant DNA. For example, bacterial DNA plus human DNA. You can see here bacterial DNA plus human DNA for the production of insulin. It is also an example of recombinant DNA. Plant DNA and bacterial DNA. Plant DNA and bacterial DNA for the improvement of crop, for the pest resistance crop production. Mouse DNA plus human DNA, that will result in transgenic animal. Okay, so recombinant DNA is nothing but the combination of two different DNAs for the production of pharmaceuticals, or increase the pest resistance of crop, or our production of transgenic animal, transgenic plant. So it is nothing but the recombinant DNA. So how recombination? So recombination is nothing but the insertion of foreign gene into the host cell, onto the host. Host means the carrier, and the carrier are different types of carriers are there. For example, plasmid. The DNA is inserted into plasmid, and then it is inserted into bacterial cell. Transformation or transfection, using the process of transformation or transfection. The organism is called as transgenic if it is eukaryote, and it is known as recombinant if it is a prokaryote. Then what is the goal of recombination? To produce the many copies or clone of particular gene. Okay, then we'll also see reporter gene. What is reporter gene? What is the use of reporter gene, etc., etc. So first of all, for the recombinant DNA technology, you will require few things. For example, the DNA of interest. First of all, you will require DNA of interest. You have to isolate that DNA. Okay. Then second thing is you will require the vector. Vector means carrier. Okay. Vector means carrier. There are few vectors, we will discuss about that. So you will require, second thing, you will require vectors. That vectors, what is the use of vectors? That desired DNA can be inserted into vector. Okay. And that vector can be inserted into that bacteria, plant, or humans for the production of desired product. For example, proteins. Okay, so there are different vectors. So what are the different vectors? The vectors may be plasmid. There are plasmids vectors. There are viral vectors like a lambda phase vector, M13 vector. There are cosmid vectors. there are particles, DNA coated with bullets. Okay, then there is a artificial chromosome or exogenous DNA. So we will see the different type of vectors here. You can see in this slide there are different type of vectors. Vector, insert sites and features are given there. Okay, so there are different type of vectors like plasmid vectors are there, then bacteriophage vectors are there, cosmid vectors are there, artificial chromosome vectors are there, then bacterial artificial chromosome, yeast artificial chromosome are the types. Okay, so first one is a plasmid vector. What is meant by plasmid? So plasmid is nothing but the extra chromosomal self-replicating DNA that is present in the bacteria. See, bacteria has two types of DNA. First one is its genetic DNA, and second one is extra chromosomal DNA. So extra chromosomal DNA is nothing but the plasmid. That is a circular DNA. Okay, that can be used as a vector. So here vectors and their insert sites and features. This chart is important for GPAT Niper and all kind of exams, because you see, plasmid, in the plasmid less than 20 kb DNA can be inserted. Okay, in case of bacteriophage up to 25 kb can be inserted. In case of cosmid 30 to 47 kb DNA can be inserted. In case of artificial vectors you can insert more size DNA. So here plasmid consists of very less amount of DNA that can be inserted, or very less size, less molecular weight DNA that can be inserted. So remember, this plasmid can be useful for only small DNA molecule. And artificial vectors, like all artificial vector, particularly the yeast artificial vector, that can be that can we can insert 100 to 1000 kb gene or DNA in that vector. So it consists of highest amount of DNA can be inserted into the yeast artificial vector. Remember this, highest molecular weight DNA or highest molecular weight outside or transgene can be inserted into yeast artificial vector, whereas smallest amount can be inserted into plasmid vectors. Okay, then bacteriophage means what? When virus will attack to the bacteria, that will result in bacteriophage. Okay, then cosmids are what is meant by cosmid? So cosmid are the vector. These are the combination of plasmid and bacteriophage. Combination of plasmids and bacteriophage will result in cosmids. And these are the some artificial vectors, that is plasmid artificial vector, bacterial artificial vectors, then yeast artificial chromosomes. So these are also some made using these plasmid and bacteriophage, but they will have additional cost side. Okay, so these are important. So what is the examples of plasmid vectors? For example, PBR 322, then PUC 19. Here I will resume it. Here P indicate the plasmid, B indicate the bacteria. Okay, R indicate the strain, and P indicate the plasmid, BR indicate the name of scientists, and 322 is the name of that species. In the similar way, PUC 19. So what are the feature of plasmid? Replicate independently on the microbial chromosome, so many copies can be maintained in the single cell. Okay, so bacteriophage is nothing but the virus attack the bacteria. When virus will attack the bacteria, it will result in the bacteriophage. So examples are lambda phage vectors, M13 vectors, etc, etc. Okay, they can be modified. Then cosmid are made by above two. It is a combination of plasmids and bacteriophage will result in cosmids. Okay, just remember here, yeast artificial vectors can be inserted with highest amount of DNA, and plasmid can be inserted with lowest amount of DNA. Okay, this is important for GPAT. Then what are the characteristics of a vector? What is the ideal characteristic of vector? Okay, so it must have origin of sequence or ORI sequence, that is also called as ORI sequence. Okay, so it can replicate independently and it must have origin of sequence, origin of replication, which is also known as ORI sequence. Then it has a restriction site. See, the your vector is nothing but the circular DNA, so you have to cut, and every restriction enzyme will act on it, so it must have restriction site for to be cut. Okay, then it must have cloning site or cloning region or multiple cloning sites. Okay, it must have reporter gene that will announce its presence in the host cell. Okay, so reporter genes may be antibiotic resistance, tetracycline resistance is there, then penicillin resistance is there, ampicillin resistance is there in a small size in comparison to the host chromosome for ease of isolation. Okay, so we will discuss what are the plasmids and how we can modify plasmid. So what are the restriction enzymes and their roles? Then origin of restriction enzymes, what is the origin of this, how restriction enzymes have identified, what is the origin of restriction enzyme. Then we will have a closer look at restriction enzyme, and then we will try to understand plasmids using the diagram. Okay, so what are the plasmid? Plasmid are nothing but the circular DNA which are extra chromosomal, self-replicating. So self-replicating extra chromosomal DNA is known as plasmid, which are represented bacteria. You can see in this slide, you can see here is a bacteria, here is a two type of genes. Here is a coiled gene that is known as genome of bacteria, that is the original gene, and this is a circular gene which is known as plasmid. This is present in naturally in the each bacteria. So you have to isolate this plasmid. This plasmid consists of one unique feature, that is self-replication. So they undergo self-replication. Okay, so plasmids are nothing but the circular DNA, extra chromosomal circular self-replicating DNA molecule. Okay, so circular DNA that is used by bacteria to store the genetic information. Modifying the plasmid includes extra genes allows for the production of new proteins. Okay, so these are the plasmid. How we can modify the plasmid? So we can modify the plasmid using restriction enzymes. We can modify the plasmid using DNA ligase enzyme. So what will restriction enzymes do? So restriction enzymes will do the cuts on that circular DNA, so that new DNA can be inserted in that. So various type of restriction enzymes are there. So here for example there BAM HI, HIND, HIND HI, HIND 3, BAM HI, HIND 3, etc. What will they do? They will cut the DNA at specific sequence, that is known as sequence. Okay, different restriction enzymes isolated from different sources, and they will have different actions. They will have different cutting action at different sites. They will have different restriction sites. Then DNA ligase, as I have already told you, the ligase enzymes are used to join the slices of DNA. So here is the important chart. Here are some restriction endonucleases and their recognition sequence. You will get this chart in the textbook of biotechnology by C.K. Dubey. Okay, so we will discuss only few. You can remember easily these kind of restriction endonucleases and their source. For example, most commonly used is EcoRI. Eco stand for E. coli and RI for restriction endonuclease. Okay, so EcoRI. So BAM HI is isolated from Bacillus amyloliquefacens H. Okay, so BAM for Bacillus, Eco for Escherichia coli, HA for Haemophilus, then HIND for Haemophilus influenza. So you can remember these enzymes using these kind of tricks. AL means Aerobacterial luteus. Okay, so these are the enzymes, restriction enzymes, and their microbial source, and this is the recognition sequence. Means, for example, E. coli restriction enzymes will cut the DNA at this sequence. If this sequence is present in that circular DNA or linear DNA, E. coli will cut the DNA at this sequence only. Okay, the BAM HI will cut the DNA at this sequence. It will not cut at this sequence. Okay, so my point is that every restriction enzyme has different recognition sequence, and they will cut at only specific recognition sequence. Okay, so what is the origin of restriction enzyme? What is the how they have identified? So we will see that. You can see virus will attack on bacteria here. So virus will attack on bacteria. After attacking, they will release their genetic material, that DNA, into the bacterial cell. Okay, so that is a foreign DNA. So bacteria produce restriction enzymes to protect against the invading viral DNA or RNA. So you can see the injected DNA is destroyed by restriction enzymes. This coiled structure inside the bacterial cell wall is nothing but the restriction enzyme. So they used to cut the DNA molecules. You can see again. So bacteria will come, it will attach on bacteria, virus will come attach bacteria, it will release the genetic information. If genetic information is if genetic information is replicates inside the bacterial cell wall, that will destroy the bacteria. So bacteria will produce the restriction enzymes to cut that DNA and make the DNA ineffective. Okay, so this is the restriction enzymes. So restriction enzymes that will cut DNA at only particular sequence. For example, EcoRI restriction enzyme of isolated from E. coli that will cut at this site. You can see in the image. This is the site. If this sequence is present on the DNA, EcoRI will cut down the DNA at this place, and the cut DNA will result in this kind of structure that is known as blunt edge or sticky edges, so that here other DNA can be inserted and they can be fused. So different restriction enzymes have different recognition sequences. This makes it possible to create wide variety of different gene fragments. Okay, so we will see the action of restriction enzyme using animation. So you have a double stranded DNA here. So restriction DNA stand with EcoRI restriction site is, so restriction site is highlighted here. So how restriction enzyme will act on it? So EcoRI, that means restriction enzyme from isolated from E. coli, will act on this site. Okay, so it will cut the DNA, which result in sticky ends. Okay, then DNA ligase, if we use DNA ligase, the function of DNA ligase to join the sticky ends. Okay, so DNA ligase will join this cut DNA. Okay, so this demonstrates the functioning of DNA ligase and restriction enzymes. So sticky ends cut with different restriction enzymes will not bond together. If we use different restriction enzymes, for example EcoRI, then BAM, if we use two different enzymes for cutting the molecule, then it will cut at different sequence, and they will not bond together because they has a different restriction site, because the base pair of sequence of two sticky ends will be different and they will not match up. Okay, so this is a ideal structure of plasmid vector. So it is an example of plasmid vector. You can see here it will have a different region. It is a PUC19 vector. It will have ORI means origin of replication. So it will have site for origin of replication. Then it will have site of lag Z. Lag Z means it is a restriction site. Restriction site means whatever are the restriction enzymes, they will act on this side. They will cut the this circular DNA at this side. Okay, here you can see hind 3 will cut at this side, BAM eqri will cut that side. Okay, then this plasmid vector also consists of some marker gene, ampicillin resistance. You can see here AMPR, that is ampicillin resistance gene is inserted into this plasmid, so that after this plasmid is injected in the bacteria multiplication, and if we isolate that bacteria in medium and allow it to grow, then how can we identify whether bacteria is transformed or not? So in that case we use penicillin antibiotic or ampicillin antibiotic in the medium. Okay, in that condition only transformer bacteria, those bacteria which consist of ampicillin resistant genes, will grow on that medium, and it will be easy for screening whether bacteria is transformed or not. We can easily identify that. So that's why the marker gene must be present in the that your plasmid or vector. So vector consists of three, that is origin of sequence, then your restriction site, then your antibiotic resistance site, and also there is one more site that is known as linkers adductors sites, and rest of the site you can insert the desired DNA. Okay, so this is the ideal structure of plasmid. So how we make recombinant DNA using restriction enzyme. Okay, so you can see circular DNA here. This is nothing but the plasmid which is isolated from bacteria. You can see here linear DNA or DNA to be inserted. Okay, so restriction enzyme will act on both. So it will cut down the sites. Whatever is marking is given in the red color, it will cut at that site. So it will result in like this. Okay, then DNA ligase will join this cut DNA like this. Okay, it will result in recombinant plasmid. Okay, so this will be recombinant DNA. So you can this kind of recombinant DNA, many possible recombinant DNA plasmid can be produced in this way, but this was desired plasmid for the experiment. For example, you can produce lot of DNA like this recombinant DNA. Okay, then you have to, whatever you have made recombinant DNA, you have to insert into the bacterial cell. Okay, for the expression means replication, transcription and expression means protein synthesis. So आपको जो प्रोटीन लग रहा है वो आपके बजाय बैक्टीरिया सिंथेसाइज करेगा. कैसे? जो प्रोटीन लग रहा है उसके लिए जो जीन रिस्पॉन्सिबल है उसको कलेक्ट करो, उसके बाद बैक्टीरिया से प्लाज्मिड कलेक्ट करो, प्लाज्मिड में वो जीन इंसर्ट करो, और वो प्लाज्मिड जो बना है रिकॉम्बिनेंट प्लाज्मिड वो बैक्टीरिया में इंसर्ट करो. उसके बाद वो प्लाज्मिड इंसर्ट होने के बाद वो वहाँ पे रेप्लिकेट हो जाएगा, वहाँ पे ट्रांसलेट हो जाएगा, एक्सप्रेस हो जाएगा और प्रोटीन सिंथेसाइज करेगा. और बाद में आप वो बैक्टीरिया को मल्टीप्लाई करके फर्मेंटेशन करके बहुत सारे बैक्टीरिया बना सकते हो. वो बैक्टीरिया को ब्रेक करके जो प्रोटीन आपको चाहिए था वो आइसोलेट कर सकते हो. ओके, सो दिस इज नथिंग बट द रिकॉम्बिनेंट डीएनए टेक्नोलॉजी. ठीक है. सो प्लाज्मिड इंसर्शन कैसे होता है? तो प्लाज्मिड है, ये बैक्टीरिया है, उसमें आपको इंसर्ट करना है, वो तो इंसर्ट हो जाएगा, ऐसे इंसर्ट हो जाएगा. वो इंसर्शन के लिए डिफरेंट मेथड्स रहते हैं. फॉर एग्जांपल यूज ऑफ डीएनए यूज ऑफ एंजाइम्स यूज करते हैं. उसके बाद जीन गन यूज करते हैं. इलेक्ट्रोस्टैटिक ट्रांसक्रिप्शन होता है, ट्रांसफेक्शन होता है उसमें. तो उसके टाइप है वो. यू कैन रीड इन द बुक. देयर आर डिफरेंट मेथड्स टू इंसर्ट द प्लाज्मिड इनटू द बैक्टीरियल सेल. ओके, so after insertion the bacteria is already to grow, colonize. They will colonize. Okay, they will make a colonies. So how you can identify this bacteria, whether it is the bacteria is our interest or not? Because with the naked eyes you cannot see whether which bacteria is transformed or which bacteria is not transferred. So that's why we have marker gene in our plasmid. Okay, so plasmid, recombinant plasmid transformation into bacterial cells. The transformation can be done by electroporation. Natural methods are also there. Then gene gun are also there. Then enzymatic methods are also there. So it will be transferred into bacteria. Some bacteria will take the plasmid, some bacteria will not take the plasmid. So you have to grow those bacteria in the medium containing antibiotics, so that those bacteria who have transformed or taken the plasmid, they will grow on it, and those who have not taken the plasmid, they will not grow on it, because it consists of penicillin or any other antibiotics. Okay, the restriction fragments will let randomly producing the plasmid forms. Bacterial insertion would be necessary. Then colony growth and other listing isolate bacteria will be desired. Plasmids. So we can use this technique for production of insulin. For example, you have to isolate the human cells and grow into the tissue culture. Then isolate the DNA from human cell. Okay. The next step is isolated DNA. You have to cut with restriction enzymes. Then these cutted DNA or small piece of DNA you have to insert into plasmid, which is you have to meanwhile isolate. You have to insert into plasmid. Then this plasmid you have to insert the DNA of interest into the plasmid. Okay. Then this recombinant plasmid you have to insert into the bacterial cell again, and you have to screen the bacteria. You can allow the bacteria to grow. Bacteria will grow, will multiply, it will make trillions of insulin producing bacteria. Okay, so all these will be transformed bacteria. Now then you can collect this bacteria for fermentation. After fermentation you can break down the cell and isolate the protein, for example your insulin. So it is the one of the application of recombinant DNA technology for the production of insulin. Okay, so it is a overview of gene cloning. It is a summary or overview of gene cloning. We will see. So human gene of interest, okay, that is responsible for the photosynthesis is isolated, that is cut using restriction enzymes. Simultaneously you have to isolate plasmid from bacteria. Then you have to insert interested gene into the recombinant plasmid. Then this plasmid will you have to transform into bacterial cell wall. Then bacterial cell wall, they will replicate, they will produce lot of bacterial colonies. Then you can isolate the desired protein, okay, after expression. So what is use of recombinant DNA technology in the farming? So farming is the production of pharmaceuticals in the animal engineered to contain foreign or drug producing gene. For example, here you can see here these are the goats, in which they are recombinant goats. In these goats there is a gene responsible for production of responsible for the dissolving of clot. So these goats consist of human gene for clot dissolving protein that is produced in their milk, so that their milk consists of drug. Their milk is nothing but the drug that is used to dissolve the small clot. For example, in case of your stroke, etc., coronary artery disease, etc. So what are the different applications in the pharmaceuticals? So recombinant DNA technology you can use for the production of insulin, as we have seen in the previous slides. Then it is used for the production of factor 8, blood clotting factor 8, okay, for the patient suffering from hemophilia. Hemophilia is the condition in which there is no the person is not able to clot the blood. There is no clotting of blood. Welcome back. So what is the use of recombinant DNA technology in the pharmaceuticals? So it is used for production of insulin, which is useful for patient treating the diabetes. Okay, patient treatment of diabetic patients. So factor 8 for the patients of suffering from hemophilia. Then also factor 9 also used for treating the patient of hemophilia B. Then you can produce growth hormones with using recombinant DNA technology, which are have wide application, like in case of dwarfism. Then erythropoietin is used for treating the anemia. Then you can produce interferons using recombinant DNA technology. What are the interferons? Interferons are nothing but the uh, they are produced they are produced by the body in the response of infection of virus. Okay, normally you see, if we got infection of bacterial infection, our body will produce antibodies. Okay, our body will produce antibodies, but in case of virus, that antibodies are known as interferons. So if you got viral infection, your body will produce interferons. So recombinant DNA technology also used for production of interferons. Then several interleukins can be produced by RDN technology. Then granulocyte macrophage colony stimulating factor, that is produced by RDNA technology, that is used for stimulating the bone marrow after bone marrow transplant. After bone marrow transplant, what happens? As a transplant is a foreign transplant, so it will not accept and it will not start the normal process. So for stimulation of that transplant, we use granulocyte macrophage colony stimulating factor. Then tissue plasminogen activator, adenosine deaminase, angiotensin, endostatin, parathyroid hormone, hepatitis B surface antigen vaccines, and diagnostic. So these are the few applications of our DNA technology in the pharmaceuticals. So embryonic cell, as I have already told you, these are the embryonic stem cells. So these can be used for the treatment of genetic treatment of various types of diseases. Okay, so stem cells are the cells, embryonic stem cells are the cells which can regenerate into whole organ. So these cells have unique property that they can regenerate whole organ. Remember only this thing. Okay, so stem cells, if exposed to proper environment, they can produce various type of tissues and organs. For example, you can see in this image, their stem cell, if exposed to this insulin thyroid hormone, it will result in adipocytes. If exposed to retinoic acid, it will result in neuron, interleukin, macrophages, like this. So stem cells can be regenerated into whole organ. Okay, stem cell therapy can be used. For example, if you affected with some neurological disorders, your neuronal growth has affected, then you can use stem cell therapy to regenerate your cells. If your brain cell has died, some few brain cells, for example, your brain cell has died, then you can use stem cell to regenerate that cells. Okay, then application of recombinant DNA technology in the fingerprinting. For example, that is mostly used in the forensic science to identify the accused. Okay, so they can isolate the DNA, multiply that DNA using polymerase chain reaction, and they have different blotting techniques. Then separation techniques using agarose electrophoresis, gel electrophoresis, polyacrylamide gel electrophoresis is there. So there are different gel electrophoresis. They can identify, match such type of DNA and easily identify the accused. It will be easy to identify accused using DNA fingerprinting. You can even using recombinant DNA technology, you can create gene libraries, etc., etc. This is the technique of gel electrophoresis. You have to pour the mixture of DNA fragments on that gel or glass plate. Here you have to pour your mixture of DNA. Then DNA mixture will be separated according to their molecular weight in between that applied current. Okay, so you will get, then you have to isolate the DNA using blotting techniques, etc., etc. That is the beyond the scope of this lecture. So genetically modified foods are also available, but that is not most second thing, how we can produce transgenic plant and how we can use RDN technology for transgenic plants. Okay, so in US, in India also there are a lot of transgenic crops. For example, BT cotton is there, golden rice is there, lot of even you can increase the size of potato. You will have a different kind of brinjals. All these are the result of DNA technology, right? Recombinant DNA technology. So here we use different vectors. In case of humans, we use humans and microbia. We use plasmid, phase vectors, different kind of vectors, cosmids, then artificial vectors. But in case of here in plants, we use two different vectors. First one is Argobacterium tumefaciens. This is the bacteria, Argobacterium tumefaciens, the bacteria which causes disease into the plant. It causes disease whose name is crown gall disease. So Argobacterium tumefaciens will cause crown gall disease. This is important for Niper. Okay. So remember this, Argobacterium tumefaciens will cause crown gall disease. So, and then there is also rhizogenous, one bacteria, that is rhizogenous, that will cause hairy root disease. So these two are important for Niper. So you must remember that Argobacterium tumefaciens will cause crown gall disease. That means tumors, large tumors on the trunk of that plant. Okay, you can easily get the images and information in the books. Okay, then rhizogenous species of bacteria will cause hairy root disease. So here this Argobacterium tumefaciens will have plasmid that is known as TI plasmid. You have to isolate that plasmid. In that we have to cut using restriction enzyme. You can insert desired gene. Okay, so this is desired gene. For example, it is a BT genes. BT genes means bacterial toxin genes. Means it will protect the plant against the different pests. So you have to insert that gene into the TI plasmid, then recombinant TI plasmid will be there. You have to introduce recombinant TI plasmid into bacteria. Again, you have to allow the bacteria to infect the plant, so that there will be transfer of gene. There will be transfer of desired gene into that plant. So this is the technique for the plant. So in this manner they have developed the golden rice. This is particularly important for Niper. They used to ask the questions on this. So you can see here are two types of rice grains. One is white and second is yellow. So yellow rice, or that is known as golden rice, it is a product of recombinant DNA technology. And why there is a need to develop such rice? Because golden rice is enriched with vitamin A. गोल्डन राइस में विटामिन ए होता है, जो कि नॉर्मल राइस में होता नहीं, या फिर अगर होता भी है तो बहुत कम क्वांटिटी में होता है, तो उसके लिए उससे माल न्यूट्रिशन होता है. सो दैट्स व्हाई वी यूज रिकॉम्बिनेंट डीएनए टेक्नोलॉजी फॉर प्रोडक्शन ऑफ विटामिन ए रिच्ड राइस that will result in the golden rice. Okay, remember this. So this is the genetically modified cotton, BT cotton. So it is a genetically modified cotton, BT cotton, to prevent the crops from the pests. Okay, so gene therapy is also there. Gene therapy is nothing but you have to deliver the correct gene. If you are suffering from any genetic disease, you have to clear your gene, then you have to isolate and correct the gene. That is nothing but the gene therapy. Okay, so there are two strategies: primary, ex vivo, in vivo gene therapy. Ex vivo means you have to isolate gene, modify the gene outside the lab, outside the body in the lab, and then you have to inject the gene into the patient for the correction of problem. And in vivo means you have to inject the gene directly into the patient. So ex vivo gene cells from deceased person are removed, then they are treated in the lab using techniques similar to bacterial transformation, as we have already seen. Then finally they are reintroduced into the patient. For example, you can see in the image, and it is more effective than in vivo technique. Transfection is the introduction of DNA into animal, plant, etc. etc. So in vivo gene therapy, introduction of gene directly into the tissue organ without removing the body cells. Okay. So if you have to remove your gene from other part, then you have to deliver the directly into the tissues. Here we use the vectors for the introduction of new gene into the body. So most of the vectors are of viral origin. So adenovirus, there are different viruses used in the transfer of genes directly to the body. So adenovirus, adeno-associated virus, then retrovirus is there, then herpes simplex virus, etc. They are used to directly transfer the gene into. Obviously, the virus is going to infect. That's why the virus is already modified. So its bad properties or properties responsible for the causing the infection, they are removed and then they can be used. Otherwise, instead of clearing the problem, it will increase the problem. So there is alternate therapy also. Liposomes, we can introduce your gene into, enclose your gene into liposome, and liposome can be used as a used for the transfer of gene. Then gene gun is there. Okay, gene gun is there. You have to just use gene gun to transfer the gene. So there are different genetic diseases. Recombinant DNA technology is used to cure the genetic disease. For example, genetic disease because of mutation or because of faulty single gene. Examples like cystic fibrosis, Huntington's disease, Tay-Sachs, hemophilia, sickle cell disease. These diseases can be treated like phenylketonuria, can be treated using that your recombinant DNA technology or gene transfer mechanism. That is also known as gene therapy. These are not so important. That's why I'm not discussing it all. So this was the your part of recombinant DNA technology, and hope you have understood it. And for more information, you can read these chapters from your the book, that is textbook of biotechnology by C.K. Dubey. Or don't worry, I will give all the PPTs to you. Here in these two lectures, what is important for GPAT Niper Drug Inspector Pharmacist? In last 10 minutes, I would like to say that. So first of all, your polymerase chain reaction is important. The steps of polymerase chain reaction and the enzyme used in that and its source, that is important. Different enzymes used in recombinant DNA technology, these are important. Different endonucleases are important. Okay, so these are important. Then properties of your vectors, different vectors are important in the GPAT perspective. Then size, different type of vectors and their respective size is important for GPAT perspective. Okay, then gene therapy is there. Then there is one more point in your syllabus, hybridoma technology. That is very simple thing. That is nothing but the fusion of two cells. Hybridoma technology is nothing but the fusion of two cells. So two cells can be fused when where if there is no cell wall. You have to remove the cell wall. If you remove the cell wall from the cell, that you can call it as a protoplast. Okay, so protoplast and they can be fused to each other, and the fusionogen used is a polyethylene glycol. You have to use polyethylene glycol as a fusionogen, and they can be fused to each other. They can transfer genetic material to each other. Their genetic material will be recombinant. Okay, so that is one point that is also beyond the scope of this lecture. So this was the your overall recombinant DNA technology and their application. Hope you have understood it, and if you have any doubts, you can contact either Rushikesh or contact either me. And don't worry, you will get the presentation of these slides.